Good day, this is Professor Will, CBMD PhD, case Sunday, June 23rd, 2019. It's 12:35:49 seconds Eastern Standard Time. And this is a continuation of my scientific reading. So let's hop to it here. Scientists make single-cell map to reprogram scar tissue in healthy heart cells. Date June 20th, 2019. Source University of North Carolina Healthcare. Summary: annually about 790,000 Americans suffer a heart attack, which leaves damaged scar tissue on the heart and limits its ability to beat efficiently. But what if scientists could reprogram scar tissue cells called fibroblasts into healthy heart muscle cells called cardiomyocytes? For the first time, researchers developed a stable, reproducible, minimalistic platform to reprogram human fibroblast cells into cardiomyocytes. Researchers have developed a stable, reproducible, minimalistic platform to reprogram human fibroblast cells into cardiomyocytes. And by taking advantage of the latest single-cell technologies in the mathematical simulation, the researchers have produced a high-resolution molecule roadmap to guide precise and efficient reprogramming. Researchers are confident their interdisciplinary approach of combining biological experiments with single-cell genomic analysis will inspire future crucial steps toward understanding the nature of human cardiomyocytes and translating this knowledge into regenerative therapies. To increase efficiency, they performed a screen of supplementary factors and identified MIR-133, a small RNA molecule that, when added to the 3-genome cocktail and with further and culture modifications, reprogrammed human cardiac fibroblast cells into cardiomyocytes at an efficiency rate of 40 to 60%. Next, the group sought to answer how the process of converting cells into cardiomyocytes actually works. To answer this, they looked at the molecular changes of individual cells throughout the process of reprogramming. Their analysis identified a critical point during the reprogramming process when a cell has to decide between progressing into a cardiomyocyte or regressing to their previous fibroblast cell fate. Once that process begins, a suite of signaling molecules and proteins launch the cells onto different molecular routes that dictate their cell type development. Researchers also created a unique cell fate index to quantitatively assess the progress of reprogramming. Using this index, they determined that cell cardiac reprogramming progresses at a much slower pace than that of previously well-described mouse reprogramming, revealing key differences across species and reprogramming conditions. You can think of this cell reprogramming process like map routes on Google. The starting scar-forming fibroblasts are like cars looking for the right GPS instructions to get them home to a cardiomyocyte fate. Our work identified roadblocks, wrong exit ramps, and gas stations, the genetic facilitators to get the fibroblast to the destination we want. Our newly developed cell fate index is like a gauge on a dashboard predicting how far home is. This work eliminates previously unidentified characteristics of human cardiac reprogramming and provides new research tools to better understand the process of cell fate transition and reprogramming within humans. Our single-cell pipelines and new algorithms can certainly be used for studying other biological processes, including differentiation, dedifferentiation, or drug response of a cell. This research approach is not limited to the heart, but heart disease remains the #1 killer in the world, and the main focus of our research. Good day, this is Professor Will, CBMD PhD, today Sunday, June 23rd, 2019. It's 13:05:32 seconds Eastern Standard Time, and this is a continuation of my scientific research. Let's hop to it here. Low-carb diet may reduce diabetes risk independent of weight loss. Researchers report reversal of metabolic syndrome in some cases. Date June 20, 2019. Source Ohio State University. Summary: a low-carb diet may have benefits for people at risk of developing type 2 diabetes even if they don't lose any weight, a new study suggests. Researchers at the Ohio State University want to know what happens to obese people with metabolic syndrome, a precursor to diabetes, when they eat a diet low in carbohydrates but don't shed any pounds. They found that more than half of study participants no longer met the criteria for metabolic syndrome immediately following a 4-week low-carb diet. The new study included 16 men and women with metabolic syndrome, a cluster of factors, and also put people at higher risk of heart disease and stroke. The conditions that contribute to metabolic syndrome include high blood pressure, high blood sugar, excess body fat around the waist, and abnormally low good HDL cholesterol or high triglyceride level. About a third of American adults have the syndrome according to the American Heart Association. After eating a low-carb diet, more than half of the participants, 5 men and 4 women, saw their metabolic syndrome reversed, even though they were fed diets that intentionally contained enough calories to keep their weight stable. Previous work in the Ohio State lab and elsewhere has shown that low-carb diets can be beneficial for people with metabolic syndrome and diabetes. But nutrition scientists and others have debated whether that's a product of the diet or a product of the weight loss typically seen when people reduce carbs. There's no doubt that people with metabolic syndrome and type 2 diabetes do better on low-carb diets, but they typically lose weight, and one of the prevailing thoughts is that the weight loss is driving the improvements. That was clearly not the case here. Our view is that restricting carbs even without weight loss improves a host of metabolic problems. Obviously, quality of diet matters because quantity is locked down in this experiment. Over about 4 months, each study participant ate 3-month-long controlled diets, high carb, moderate carb, and low carb, with a 2-week break between diets. The order in which the participants ate the diets was randomly assigned. The research team led by research scientists ensured that the participants would not lose weight by providing them with pre-prepared meals that contain an amount of calories equal to their energy expenditure. After eating the low-carb diet, the participants had a variety of significantly improved health measures, particularly lower triglycerides and improved cholesterol readings. Despite the fact that the low-carb diet contained 2.5 times more saturated fat than the high-carb diet, it decreased saturated fat in the bloodstream. It was associated with an increase in the size of cholesterol particles in the blood, which decreases the risk of cardiovascular disease. The researchers also reported evidence of increased fat-burning efficiency after a low-carb diet and an improvement in blood sugar. They did not see statistically significant improvements in blood pressure or insulin resistance. 3 participants no longer had metabolic syndrome after the moderate carbohydrate diet, and 1 no longer had the syndrome after the high-carb diet. Researchers said that those results are likely explained by the fact that even these study diets, particularly the moderate carb diet, represented a shift toward fewer carbs for study participants. Even a modest restriction as carbs is enough to reverse metabolic syndrome in some people, but others need to restrict even more. Because of the study design, waist circumference was not factored in as a contributor to the metabolic syndrome. Had the participants been permitted to lose weight, it is likely that several more would have been considered free of the condition after the low-carb diet. This research doesn't address the potential long-term benefits and challenges of adopting a low-carbohydrate diet, and the researchers suggest that future long-term diet studies on people with metabolic syndrome need to include low-carb diets. Good day, this is Professor Will, CBMD PhD. Today, Sunday, June 23rd, 2019. It's 13:39, 40 seconds Eastern Standard Time. And this is a continuation of my scientific research, so let's hop to it here. Inhaling air pollution like irritant alters defensive heart-lung reflex for hypertension. Date June 18, 2019. Source University of South Florida, USF Innovation. Summary: using a rat model for high blood pressure, hypertension, a common chronic cardiovascular condition, research found that pre-existing hypertension altered normal reflexes in the lungs to affect autonomic regulation of the heart when an irritant mimicking air pollution was inhaled. In healthy people, inhaling ozone or particle pollution triggers a defense lung heart reflex pulmonary cardiac reflex that automatically slow heart rate to accommodate oxygen deficiency and help slow distribution of pollutants throughout the body. Yet, when patients with cardiovascular disease breathe pollutants, that some protective mechanism does not kick in. Instead, their heart rates intermittently speed up, known as tachycardia, and can evoke a potentially deadly irregular heart rhythm, known as premature ventricular contraction. Their preclinical findings reported May 11th Journal of Physiology help explain the altered physiological response to air pollution in patients with pre-existing cardiovascular disease. Using a rat model for high blood pressure hypertension, a common chronic cardiovascular condition, the USF health team found that pre-existing hypertension altered normal reflexes in the lungs to affect autonomic regulation of the heart when an irritant mimicking air pollution was inhaled. In particular, hypertension appeared to shift the reflex response from the parasympathetic nervous system to a sympathetic nervous system. The sympathetic nervous system mobilizes the body's defense fight or flight response to a threat, including releasing adrenaline that increases heart rate. In contrast, the parasympathetic nervous system controls involuntary responses, including breathing and heart rate, while the body is at rest and maintains a state of calm. Speeding up of heart rate and abnormal heartbeats in the hypertensive rats were due to the switching on of this flight or fight nervous system not seen in the healthy animals exposed to noxious agent. The heart was responding to an aberrant nerve-generated reflex that may worsen pre-existing cardiovascular disease. To simulate effects of air pollution inhaled into the lungs, difficult to recreate in a laboratory setting, USF researchers used aloe, isothiocyanate, the pungent ingredient in wasabi, and horseradish. When healthy rats with normal blood pressure inhaled this irritant, their heart rates slowed as expected. Within the rats with chronic hypertension, inhaling the same irritant stimulated an increased heart rate accompanied by premature ventricular contractions. Surprisingly, a rapid heart rate and abnormal heart rhythm did not occur when aloe isothiocyanate was intravenously injected into the hypertensive rat. It did not evoke the peculiar reflex. Instead, we observed a slowing of the heart rate like that seen in the rats with normal blood pressure. This suggests that the sensory airway nerves accessible by IV are different than those accessible by inhalation. So perhaps the pathway of airway sensory nerves connecting organs like the heart and lungs with the brainstem, are more complex than previously understood. Chronic hypertension may remodel airway sensory nerves controlling the pulmonary cardiac reflex that helps defend the body against physical damage from airway pollution, the USF study suggests. This remodeling, of which may happen in the developmental stages of hypertension, could turn on inappropriate sympathetic nervous system excitation of the heart, the researchers say. By better understanding how cardiovascular disease changes neuronal interactions between the heart and lungs, the researchers hope to help doctors with treatment choices and eventually discover new treatments. The researchers' goal is to add another piece of information that clinicians could consider when selecting a best treatment for hypertension, in addition to the patient's age, ethnicity, and race. That might include whether the person lives in an area with high pollution levels. In the long term, if we can identify the nervous system mechanisms involved in the remodeling of pulmonary cardiac reflex, we can target those to develop new blood pressure drugs. Good day, this is Professor Will, CBMD PhD. Today, Sunday, June 23, 2019. It's 14:41, 15 seconds Eastern Standard Time. And this is the end of this module. Hope you enjoyed listening. Have a great day.